Hello students, welcome back to NEET World channel. This is Narayani, I am your botany educator. So in previous class we already completed the nitrogen cycle students. So now see the biological fixation. So before we are getting to the topic, I would like to inform very important news for all of you. So here we have started NEET special program for just rupees 3,333 per month. It is especially for the offer for 12th and 11th and 12th students and also who are completed their 12th. If you are really passionate to become a doctor with affordable fee, we are providing this is the best platform for you. You are going to get the benefits like live plus recording sessions along with well crafted study material is also provided. If you are really passionate, if you regularly go through our classes and if itself you do your well crafted study material, far more enough to get good rank in your upcoming examination for sure. So limited seats are available students, grab this opportunity as soon as possible. Got it? If you have any other queries, please let me know by this number. It is available both offline as well as online. As per your convenience, you can choose the either offline as well as online. And one more important news students. So by scanning this QR code, you can join in our Telegram group. So in this Telegram group, we are providing the digitalized plus handwritten notes. Along with that, we are providing daily quizzes of botany, physics, biology and also geology, chemistry and also mathematics. For any competitive examination students, it is doing of mocks is very important. So you will get in our Telegram group. And live classes freely information is given to the in our Telegram group, and also whatever the free resources will be posted there, and mainly if you have any doubts you can ask directly in the Telegram group. So by the concerned lecturer can get back to your doubt and your doubt will be solved. Got it? If you have any further more queries to inquire about us, please contact this number. So let's dive into the topic of today's students. So we have completed the nitrogen cycle. So in nitrogen cycle students, what are the steps are involved? That is, first one is nitrogen fixation. First one is nitrogen fixation. It can be happen by the natural methods as well as the biological fixation methods also. As by the natural methods, it can be done by the lightning, and also in the industrial process there is a Haber's process will be done, right? So through that it is happening, and another one is biological fixation. So to discuss in that biological fixation students, let's fastly recall the yesterday's topic. So what are the steps involved students? Nitrogen fixation. What is nitrogen fixation students? That is the conversion of nitrogen into forms of ammonia and ammonium, right? The next step is ammonification. What is ammonification students? The conversion of organic proteins from dead plants and animals into ammonium compounds is nothing but ammonification, right? Coming to the third one, what is that students? That is nitrification. So this, whatever the compounds of nitrification, 1 minute. So the conversion of ammonia into utilizable forms like nitrites and nitrates, right? Nitrites and nitrates that will be absorbed by the soil, and through soil the plants will be absorbed. So thereby the deficiency of the nitrogen will be cleared by the plants, right? The last step is denitrification. What is the denitrification students? So whatever the compounds like nitrates, they will be formed to the original form nitrogen through the bacteria. That bacteria we call it as denitrifying bacteria, right? So that is why the stable in the atmosphere there is the nitrogen present at the stable of 78%. How it is getting stable? Because it is again get back into the atmosphere. These are the 4 more steps. And after nitrification there is a process of assimilation is also will be there, students. What is meant by assimilation? Incorporation. What is that, students? That is incorporation. What is incorporating? Whatever the elements, whatever the elements is observed by in the forms of nitrates and nitrites, so these will be incorporated into the plants and animal bodies. How the plants get incorporated, how it is utilizing the nitrogen compounds, right? And how it is, it is helping making of proteins, amino acids and nucleic acids, right? So this is what about the assimilation. Incorporation, which means assimilating. Assimilating is nothing but incorporating and utilizing the forms of nitrogen. We don't have that mechanism. Plants also don't have that mechanism. But plants do have that mechanism called nitrogen. Now, nitrogen compounds like nitrates and nitrites. Got it? So see, example I given you already that, so if you take the cocoa bean, will not eat directly. Why you will not eat directly? Because it is not in the utilizable form that we can eat. But in the form of chocolate, we will eat. Because it is in the eatable form, right? So like this is how the, the nitrogen is actually present in the atmosphere here. It is an inert in condition which is N triple bond N. It is very hard to break down by the plants and animals. We don't have direct mechanism for that. But we are getting this nitrogen by the compounds like nitrates and ammonia. Got it? So this is what we discussed in the yesterday's class, students. So now we are discussing how nitrogen is biologically fixation happens by the plants. How biologically the fixation happens by the plants. Let's see that. So let's see that, students. So see, here is one of the beautiful image, students. See, this is the atmospheric nitrogen. This is the atmospheric nitrogen. The nitrogen fixing bacteria, which means only few organisms can fix the nitrogen. So those organisms are nothing but bacteria. There are prokaryotics. The prokaryotics, which means the nitrogen fixing bacteria, they can fix the atmospheric nitrogen and makes into utilizable forms like ammonia and ammonium with the help of the more utilizable forms like nitrates and nitrates through the nitrifying bacteria, right? So it is absorbed by the plants. So whatever these amounts will be, nitrates and nitrates again get back to the atmosphere by the denitrifying bacteria. Ammonification is also happens by the organic material, which means the conversion of dead plants, animals. These complex organic proteins are converted into ammonia through the ammonification by the bacteria. What is the examples of that, students? Bacillus racemose, beta vulgaris, right? So some of the examples I given to you. So this is about the nitrogen cycle. So see how beautifully for one picture you can answer it is how the plants are observing. So these compounds are helps by through the roots, through the vascular system like xylem. It is exporting to the all parts of the plant. Thereby plants can make their proteins, amino acids, and it helps in the preparation of their nucleic acids. Got it? So this is how it is making up. Now see the biological fixation. Now see the students, biological fixation. So now see that one. So that is very important topic, students. So you have to be very alert by saying while listening the class. Biological nitrogen fixation. What is meant by biological nitrogen fixations? We don't have that mechanism. Plants don't have that mechanism. Very few organisms that have mechanism. What are those organisms? Very few living organisms can utilize the nitrogen, which are present abundantly in the atmosphere. We all know that microorganisms are everywhere in the they are present abundantly in the atmosphere, right. So in the handful of soil there is a more than presence of hundreds of bacteria, right. Bacteria is more abundantly present in the atmosphere. Only prokaryotic species can capable of fixing nitrogen. This nitrogen can who have that ability to fix the nitrogen, which is in the form of inert condition. In this form, who can fix it? That is that is prokaryotic organisms can able to fix nitrogen. See, so the reduction of nitrogen into ammonia by living organisms, by the important enzyme name called nitrogenase. How these prokaryotic organisms, how they are able to fix the nitrogen? Because these prokaryotic organisms have the very important enzyme name called nitrogenase enzyme in it. So they are these enzymes exclusively present in the prokaryotes. By the activation of this enzyme, these organisms can fix the nitrogen. This enzyme have ability to fix the nitrogen which is present in the atmosphere. Got it? So it is in the form of this nitrogen. Through the nitrogenase enzyme, it is converted into NH3, which means utilizable form it is converting. Which organisms that are, students? That are prokaryotic organisms, which means they are bacteria. So which type of bacteria can fix this nitrogen? So they are free living bacteria and symbiotic association bacteria are there. Free living aerobic bacteria, which means these bacteria can utilize the oxygen also. These bacteria can utilize oxygen, they are surviving. So that free living aerobic microbes, they are Histobacter, Basankia, cyanobacteria like Enabina and Nostac. And free living anaerobic, which means they are actively in the absence of oxygen. Anaerobes, which are they can abundantly grow in the absence of oxygen, right. So these are Rhodospirillum, Bacillus, Clostridium. Got it? So symbiotic bacteria, which means what is mean the symbiotic, which have the mutual benefit, which this bacteria having the mutual, which it is always in a collaboration between the two different organisms, like bacteria with the host. The host is plant. The host is, students, plant. One is bacteria, then for its host is plant. So which bacteria that are? Rhizobium and Frankia. Rhizobium and Frankia. Now see the symbiotic biological nitrogen fixation. Most prominent is legume bacteria, which means because the most plants like legume plants are making the symbiotic relationship with this bacteria. See, such as Rhizobium is a bacteria which is rod shaped, is always associated with many legume plants such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans. These are the very important example, students, that you have to be remember for sure. For your MCET point of view, you have to be remember these examples for sure. So the most the host relation will be is the bacteria and the host. The host always will be the plant. So these are the leguminous plants which always the Rhizobium bacteria associated with, and helps in making a fixation of nitrogen. Got it? Now see, so how exactly happens? Association shows with many legume plants, like these are the examples, right? So the most common association shows the nodules. What is there forming the nodules? What these are nodules? In the roots, see, the outgrowth of the roots is nothing but the nodules. The outgrowth of the roots is nothing but nodules. These are the symbiotic association. Got it? Now see, some always bacteria, most of the bacteria can associate with leguminous plants, of course, but some bacteria, Frankia, this bacteria associated with Alanus, which means it is a non-leguminous plant. It is not a non-leguminous plant. They will give like Frankia in one column and Elnus in one column, which we have to keep the match the following like the right option. Frankia is the bacteria which shows the symbiotic association with the plant called non-leguminous plant like Elnus. Got it, students? Now see, this shows the symbiotic association, which means we are now studying the rhizobium bacteria, how it is get associated. This association always happens before flowering. Always happens before flowering. You will see near the special outgrowth nothing but nodules, which means the ultimate for the fixation of nitrogen, there is a formation of nodule is required. Got it? The outgrowth has the, if you see this outgrowth, the central, the outgrowth that with the ball like structure, that we call it as root nodule. So the roots having the outgrowth called nodule, they are the ball like structures. When you see the central portion of it, it is appears like red and mostly pink. Why these are appear in these color? Because due to the presence of leg hemoglobin. We all know that hemoglobin gives the color of blood, right? So which always in the red color. As it is occurs in the infected cells, I will in the look into the further topic get to know why we are calling the infected cells. They are appearing in the pink color. So that is why they are forming the leguminous hemoglobin. Now see how exactly the formation of nodule occurs. Because of the nodule formation only the nitrogen can fix by the plants, right? So how these bacteria is responsible for the fixation of formation of nodule, and it is how it is forming the and how it is fixing the nitrogen. Because why these bacteria only can fix the nitrogen, students? Yes, because of the enzyme name called nitrogenase enzyme is present only in the prokaryotic organisms like bacteria, right? Now see, nodule formation. How exactly formation occurs by the plant? This process is not obligatory, which means it is not mandatory. Whenever the plants require the nitrogen, what will happen? This root hair is there, no? That will release the chemoattractants, which means it gives the signal to the bacteria. The bacteria is in the colonies like presence. We all know that bacteria is abundant in the atmosphere. So when this bacteria receiving that signal, so they are releasing the chemoattractants, nothing but flavonoids and betaines. So when signal is giving to this bacteria, what will happen? This bacteria get associated at get attached at this receptors. So there will be a signal releasing by this root hair, right? When the signal is releasing by this root hair, the bacteria come and attach at its receptors. At its receptors. When they attaches, the root hair is elongated and make it to allow the bacteria inside of it. Allows the bacteria to inside of it. So this bacteria, it shows the curling. So when it is curling the root hair is curling, the bacteria is getting inside into the plant. So when it is getting inside, what will happen? This bacteria, we all know that bacteria having the, its hard material called cell wall. While it is entering into the host, it loses its cell wall. And it is surrounded by the plant derived structures. It is surrounded by the plant derived structures. Then these bacteria we call it as bacterioids. What we call, students? That is bacterioids. So it is forming it is entering into the cortical cells. As it is going like the infections way, that is why we call it as infection thread containing the bacteria. Got it? It is entered into the inner cortex. So it is associated with the vascular system. So by that bacteria getting the nutrients and shelter from the plant, and the plant is fixing the nitrogen, because this bacteria is forming the root nodule. So what is the work of this nodule? it will block the oxygen. Why it is blocking the oxygen is required? Because to fix the nitrogen, the bacteria having the nitrogenous enzyme, right? The bacteria having the nitrogenous enzyme, right? That will be this enzyme is always active in the absence of oxygen only. So when this bacteria is fixing this, forming this root nodule, this root nodule will block the oxygen. So at that time, this nitrogenous enzyme will be active and there is the fixation of nitrogen is happened. Fixation of nitrogen will be happened. So it will be conversion of nitrogen into ammonia. That work will be done. So that ammonia will be utilized for the plant purposes to prepare their proteins, amino acids, nucleic acids. Got it, students? Now see the written notes. Involves in the sequence of multiple interactions, that is rhizobium bacteria and the host plant, which means leguminous plants, right? Rhizobia. These rhizobia multiply and colonies. It will be found on the soil by the colonies, right? So this surrounding the roots, when the it is, I already said it is not mandatory. When the plant needs, what will happen? It releases the signal. So when the signal, the signals are chemoattractants released by the plant. Those chemoattractants will give the signal to the bacteria. Then bacteria come and attach to the receptors present in the root hair, right? So then the root hair will be curled and take this bacteria inside of it. So that time it will appear like the infection thread, right? See, rhizobia multiply and colonize the surrounding the roots. Get attached to the epidermal root hair. It is get attached to the epidermal root hair. So this root hair curl. It will forms the curling, right? See, exactly. It is forming the curling, right? Yeah. So it formed the curl. The bacteria invade into the root, which means it is invade into the root. Now see, an infection thread is produced. Like that, what is this bacteria is getting inside? It forms the infection thread. So carrying the bacteria into the cortex of the root. It is taking the bacteria into the cortex of the root. They initiate the nodule formation. Then the bacteria which lose their cell wall and it is surrounded by the plant derived structures, it forms the root nodule in the cortex of the root. So then the bacteria released from the thread into the cells, which lead to the differentiation of the specialized nitrogen fixing cells. So when it is enters into the cortex of the root, so what will happen? Which leads to the differentiation. We all know that bacteria can easily multiply, right? So it have the in less time it can multiply its own species, right? So it will multiply and forms the specialized nitrogen fixation cells called nodule formation is done. So what is the work of that nodule? So it blocks the oxygen. Why it is need to block the oxygen, students? Because when it is binds with the oxygen, the nitrogenous enzyme present in it will be active, right? So then the nodule is formed. When the nitrogenous enzyme is activated, then it is able to fix the nitrogen. Then plants get the nitrogen in their available forms. Then it will do its work by protein making, nucleic acids, like that. Got it? Then the nodule is formed. It contains the necessary biochemical components such as nitrogenase enzyme. Nitrogenase enzyme. It will be formed by only because of the blocking of oxygen. It will always, the nitrogenase enzyme will activate in the absence of oxygen only. Got it? Now see, it establishes a direct vascular connection with the host and exchange of nutrients. We said that is it symbiotic association, the bacteria fixing the nitrogen. Then what the bacteria is benefiting? By the nutrients, as it is attached to the cortex of the vascular system, it will absorb nutrients from the plant. Got it? And it gets the shelter from it. So now this, see, we all said that it is the nitrogenous enzyme will always get active in the absence of oxygen, right? So it is having the very important protein name called molybdenum and iron, which helps in the conversion of nitrogen into ammonia. This nitrogenous enzyme having the main important element called molybdenum and iron, which helps in the conversion of nitrogen into ammonia. Got it? So it is very important question, students, for IPE point of view also. Let's see the question now. Let's see the question. So here is the given. Explain the steps involved in the formation of root nodule. Involved in the formation of root nodule. So mostly the fixation occurs between the rhizobium with leguminous plants. And also one more example I given, that is symbiotic association occurs by the Frankia plant with non-leguminous plant like Elenus. Non-leguminous plant like, students, that is Elenus, right? So here they explain the steps involved in the formation of root nodule. Here they are asking about how the root nodule is forming. How the root nodule is forming. The steps involved in the, what are the steps that we have to explain? For the IPE point of view, students, it is one of the very important question. See this question. The roots of the host legume. So whatever the host legume, so I said it is not a mandatory, right? It is not obligatory. Whenever it requires, what will the roots will do? The roots of the legume plant releases sugars and amino acids, nothing but chemoattractants. Chemoattractants. They are flavonoids and betaines. So when they given the signal to the bacteria, what will happen? These sugars, which means these chemical attractants, will attract the bacteria name called rhizobia. Bacteria name called rhizobia. It is in a rod shape. Mainly association will happens with the leguminous plants, right? What will happen? They multiply and colonies. The bacteria will always be in the rapid condition and will be in the forms of colonies, right? And get attached. They all come near to the root hair of the leguminous plants and then get attached to the epidermis of the root hair cells. Then get attached to the root. So epidermal cells of the root hairs, because they are having the receptors called lectin receptors, right? These root hairs curl. Then what will happen? The root hairs will be curl. They form the curling. So and by that curling, the bacteria is getting into the root hair. So the root hair curl and bacteria spread into the cortex of the root. Then it is slowly the bacteria enters into the root, right? So there is an infection thread is produced. That what we call, students, that is the infection thread. While going this bacterials into the root, root hair, what will happen? The bacteria loses its cell wall and it is surrounded by the plant derived structures. Then we call them as bacterioids. Then what we call, students? That we call bacterioids. It carries the bacteria into cortex. So whenever this bacteria is carried into the cortex of the root, right, these bacteria initiate nodule formation. So when it reaches the cortex, what will happen? There is the initiation of the nodule into the cortex of the root. Then the bacteria present in these cortical cells, the bacteria which is present in the cortical cells stimulates the host cells to divide. It helps to divide the host cells and lets the formation of the happen of nodule. This leads to differentiation of the specialized nitrogen fixing cells which forms the root nodule, which leads to the formation of root nodule. This new nodule, what will happen? It blocks the oxygen, then the nitrogenous enzyme will be active. So the bacteria can fix the nitrogen into the utilizable form like ammonia, right? Thus the nodule is thus formed, establishing a direct vascular connection. It is directly through the cortical cells, it will attach to the vascular system. So with the host, it exchange of nutrients. The plant is getting benefited by the nitrogen and the bacteria is getting benefited by the nutrients from the plant and also shelter. This is how the symbiotic relationship is happening by the both beneficial of the host and the bacteria. Got it, students? This is what the question of root nodule formation. This is one of the very important question, students. For your IPE, you have to remember this question. So, and one more very important news for important question for the IPE students, that is, see, write in brief how plants synthesize amino acids. So, which means after we all know that the nitrogen is the element which is required for the synthesis of proteins. The proteins are made up of amino acids only, right? How this question is how plants synthesize amino acids, right? This question is that one. So, tomorrow we will explain this question, students. Before that, so how it is happening also we will see in the next class. Keep revise this question, students. It is one of the very important question. So, for your IPU point of view and also for MSET point of view, you will get many questions here. So, almost we get end of this topic, students. I take only there is one class will remain to complete this session of this mineral nutrition chapter, students. Then we will do the bunch of MSET questions. Then you will get the confidence. Got it, students? So, this is about today's session, students. And before I am winding up the class, students, I would like to remind very important news for you, all of you. So, here we are started the NEET long term special program, students, for especially for the 11th and 12th and also who are completed the 12th. So, for the college goers, it is a best opportunity. So, by preparing along with the IPU, you can get the very good score in upcoming NEET and EAP set examination for sure. Because if you enroll our course, if you got our live plus recording sessions, along with if you do our well crafted study material mocks, it is very, it is definitely you will get a very good rank in your upcoming examination for sure. So, and you will get the one-on-one mentorship and one-on-one doubt solving sessions also will be there. So, as per your convenience, you can join with us by either offline as well as online. If you want to join online, we will give you access in our app name called NEET World. So, there will give access, students. So, you can get back to this number if you want to enroll our course. There are very limited seats. Limited seats are available, students. Grab this opportunity as soon as possible. If you want to join offline, you can come to our institute name called NEET World, present in Hyderabad, Narayani. So, if you have any further queries, please let us know by this number. Don't miss this opportunity, students, because it is very affordable fee of rupees 3,333 per month. If you are really determined, this is the best platform for sure. And another more important news, students, if you are very determined, the competitive examinations are for every competitive examination, the mocks are very important to do. So, here in this our telegram group, we are posting biology, chemistry, physics and also mathematics, the numerous MCQs we are providing. So, by joining in our telegram group, you will get numerous mock exams will be available, mock questions will be given. So, if you have any doubt, you can directly contact to the particular lecturers and ask the questions. They will clear out all the doubts. So, don't miss to join in our telegram group also. And whatever the live classes we are scheduling, that will be clearly informed to you in this telegram channel, and whatever the handwritten notes, digitalized printed materials will be posted right here. And also mainly your doubt solving sessions also will be there. It will be quite benefited to you, children. So, if you have any further queries, please let us know by this number. So, this is what about today's session, students. Let's meet the tomorrow session. Thank you.